Hello friends, welcome to my channel. I am Varsha Gaitonde and this topic is about the scientist's contribution to plant tissue culture. Here I have enlisted the scientists who contributed for tissue culture. The first scientist who started this work was 1838 and 39, Schleiden and Schwann, who gave cell theory. According to them, cells are the basic units of all living organisms and are autonomous or autonomic, which are potentially totipotent, means which can grow themselves, then physiological and developmental units. These are capable of regeneration to give a complete plant that is nothing but totipotent. In 1982, Sachs was the scientist who told that plants synthesize organ-forming substance which are polarly distributed, means he gave the direction. In 1902, Haberlandt, who is called as father of tissue culture, developed the concept of in vitro cell culture on artificial media. He cultured palisade cells, glandular hair and pith cells on a simple medium, that is NOPS solution enriched with glucose and peptone. Then cells increased in size, accumulated starch but failed to develop. But the first attempt was made by Haberlandt. In 1904, Henning made first attempt to embryo culture. He excised mature embryo from seeds of selected crucifers and successfully grew them to maturity on mineral salts and sugar solutions. Small weak plantlets were obtained by this process. In 1922, Colt and Robbins, they made in vitro culture isolated roots and shoot tips respectively, means root and shoot tip in vitro cultures they made. In 1925, Lebach, he developed a technique of embryo culture used to recover hybrid progeny from interspecific crosses in linum. Next is in 1934, White. As you know, White is popular for his White's media. He proposed continuous culture. You might have heard continuous culture, suspension culture, batch culture and all those things in tissue culture. So White proposed continuous culture of tomato on excised roots, introduced vitamin B as growth supplement and gave the first defined tissue culture medium, that is white medium. In 1934, Gotthardt successfully cultured and did prolification of several tree plants. That means Gotthardt worked with the tree species and he published the first handbook on plant tissue culture. This is very important. In any of the competitive exams, you may find this kind of questions like who is father of tissue culture or who published the first handbook of plant tissue culture, and the contribution goes to Gotthardt. Next is in 1937, that is Nobecourt, who made continuous callus culture of carrot. In 1939, Gotthardt, White and Nobecourt, they gave first continuous growing callus culture which established from cambium tissue. In 1957, Skoog and Miller, they reported that root and shoot differentiation in the culture. In 1958, Maheshwari and Rangaswamy, they made regeneration of somatic embryo in vitro from nucellus of citrus ovules. In 1959, Reinert and Stewart, they gave somatic embryo development, that is regeneration of embryo from callus clams and cell suspension in carrot. 1959 only, Braun, he regenerated first plant from mature plant cell. In 1959 only, Street, what he did, he gave the role of vitamins in root culture. In 1960, Kanta gave first successful test tube fertilization in papaver. In 1960, the same year, Cocking made first to isolate protoplast. He obtained large number of protoplast by enzymatic degradation of cell wall, means cell maceration was the contribution of Cocking. In 1960 and 64, Morel made vegetative propagation of orchids by meristem culture. developed shoot apex culture. This you need to remember. The shoot apex culture was given by Morel. Next, in 1960, Bergman, he made filtration of cell suspensions, isolation of single cell by plating. In 1961, Torrey and Reinert developed cell suspension culture in plants. This also you need to remember. Torrey and Reinert in 1961 developed cell suspension culture in plants. In 1962, Morashige and Skog, this is a very popular media, developed this media. Then in 1968, here onwards you may find different medias. In 1968, Gamberg and co-workers developed B5 medium. In 1969, Nish developed N6 nutrient medium. In 70, Power et al. Or Power and co-workers, their first achievement of protoplast fusion, then interspecific plant hybridization of protoplast fusion, and for Penunia hybrida and Petunia poroides. In 1972, Carlson and co-workers, they did interspecific hybridization of Nicotiana species using protoplasts. In 1975, Jangenbach and Green, who reported positive in vitro selection of maize gallus culture, show resistant to T-toxin of Helminthosporium maides. Then in 1976, San Neom gave ovule or ovary culture in Hordeum vulgare. This is also important. Previously it is asked in the NET exam also. So in 1976, San Neom, who gave ovule or ovary culture in Hordeum vulgare, means he worked with the female reproductive part. Then in 1979, Melchers worked with potato somatic hybridization and also with the tomato by protoplast fusion. Then in 1981, protoplast fusion is a major approach in distant hybridization. Okay, you need to remember this. Then in 1981, Larkin and Scrocraft, these two scientists are also important, introduced the term somaclonal variation. They worked with geranium and potato, then replaced the earlier terms like caliclone and protoclone. By removing these two names, they gave somaclone. Okay. Then in 1982, Fujita and co-workers, they gave chiconin is the first example of biochemical production from cultured cell plants, means they contributed for the plant biofarming. In 1983, Pelleter and co-workers made intergenic cytoplasmic hybridization in radish and rape. Remember this. Pelletier gave intergenic cytoplasmic hybridization in radish and rape. In 1984, Evans and co-workers gave the term gametoclonal variation, which introduced for the variability among pollen-derived cells or plants. In 1985, Kitto and Jenick produced artificial seed. This is also very important. You should know the concept of artificial seed. It is assignment for you. Go to the Google and search how the artificial seed is made and what is the composition of that particular seed, whether it is successful or failure as a seed. Okay. Then in 1993, Kranz gave complete plant regeneration, and he is also the contributor for Kranz anatomy. Coming to the haploid and double haploid history, the concept was first reported in 1922 by Blaxley, means haploid was first introduced by Blaxley in 1922 in Datura stomanium. In 1966, Guha and Maheshwari produced first haploid from pollen grain of Datura. Then in 1970, Kasha and Kao made hybrid embryo culture and subsequent chromosome elimination of haploid production in barley. In 1979, Burt and co-workers worked in haploid tobacco. Then in 1970, Kasha and Kao, this is repeating. Then the successful double haploids are produced in tobacco, wheat, maize, barley and rapeseed. The scientists who contributed for this I didn't get. If you get, please put it in the comment box so that it will be helpful for me. And Double haploids are produced from the haploids only by the colchicine treatment or by using inducers, so that there are no accurate records about it. That's it. Thank you. I hope you enjoyed this video, and if you liked, please do not forget to like, share, comment and subscribe my channel. Thank you once again.